I just want to go quickly over this the chapter, which is the first chapter of biochemistry. We have officially finished the biology chapters, and from now on, we can just do the flashcards and review questions on them. Anything that we don't know or knowledge gaps that we have to fill. So let's now go with the biochemistry chapter 1. So in chapter 1 of biochemistry is all about amino acids, peptides, and proteins. So we know that amino acids are found in proteins. So amino acids are molecules with 4 groups attached to a central alpha carbon. So the central carbon, alpha carbon, is attached to amino group, carboxylic acid group, a hydrogen atom, and an R group. The R group is the side chain that determines the function of that amino acid. And we know because it's an alpha carbon, it's a chiral center. So that chiral center defines the stereochemistry for that amino acid. And all amino acids are L-lamino acids. That's how I memorize it, lamino acids. And there's only one exception. I mean, 2 exceptions. So glycine is achiral because it's only 1 hydrogen. So it makes the amino acid to have 2 hydrogens, which are the same molecule, and eventually that makes it an achiral molecule compound. And cysteine, because it has the sulfur and it is in a higher order compared to oxygen that exists on carboxylic acid, that makes it a higher order. So like whatever the name was, there's a specific name for this, we'll come across it later. But yes, this thing is not L, but it's D. So yeah, and then we have hydrophobic and hydrophilic amino acids. So as the name implies, hydrophobic fears from the water, from hydrogen, and hydrophilic loves water. So amino acids with long alkyl chains are hydrophobic. So these alkyl chains uh, makes them hydrophobic, true, and they should be long, right? So long alkyl is hydrophobic, and those with charges are hydrophilic. And all others fall in somewhere in between. So for example, this is the structure of the amino acid that gets the variable side chain. Now then we have the acid-base chemistry of amino acids. So we have amphoteric, which means amino acids can act as a base or an acid. So the base part comes from the amino part, and the acid part comes from the carboxylic acid. And we have pKa, which is the pH at which half of the species is deprotonated. So HA, where the hydrogen and acid are attached together, equals the A minus, the concentration, the brackets are showing concentrations, right? So the ion concentration equals the acid concentration, so it's equal. So this is pKa, and we have pH, where lower pH implies acidic, right? So amino acid is fully protonated. Full protonation implies full protonation means it's more acidic. So protonation, protons comes from the hydrogens, so you can see hydrogens as protons. That's how we like to see them, right? They're not, you don't see them as electrons, right? So if the pH equals the isoelectric point, which is here, Pi, it means that the amino acid is a neutral zwitterion. This is important, right? And if the pH is higher than the isoelectric point, the amino acid is fully not protonated but deprotonated, right? Amazing. Now, isoelectric point, we have that it is the pH at which an amino acid is in zwitterion form. So at the isoelectric point, the amino acid is neutral. It's not fully protonated. It's not fully deprotonated. So it's a deuterion, and the charges cancel out to make a neutral molecule. So it's neutral deuterion and Pi. Now we have when you have no side chains, so there is no side chain here, we know that this should be neutral, right? One minus here, one plus there, one positive here, one negative here. So it's so for no side chain, you have the isoelectric point as half of the pKa1. So which one should be the lower pH? It should be the carboxylic acid, right? So pKa1 plus pKa2. Now for a neutral side chain, it's the same. So it's half of pKa1 plus pKa2. But for a basic side chain, when this side chain is basic, it's half of pKa2, which is the amino group, and plus the basic side chain, pKa3. So this is our third one. Again, the first one is the carboxylic acid because it's the most acidic, so the lowest pKa. pKa is all, you can see it as the pH, but it's the pH at which half of the species are deprotonated, right? So half of it is deprotonated. It's like in a balance of deprotonation, right? Now, again, carboxylic acid group is pKa1, pKa2 here, and pKa3 is the side chain. So if the side chain is basic, it's pKa2 plus pKa3 divided by 2. And if it's an acidic side chain, it's pKa1 plus pKa3 divided by 2. Amazing. Now, the titration is midpoint. In midpoint, so what is titration? We need to know what titration is. So we can quickly search about titration here. I have so many tabs open, so don't worry about it. Titration is a technique where a solution of known concentration is used to determine the concentration of an unknown solution. So we have something that is the known concentration. So then we want to make sure what is the concentration of the unknown, right? How, for example, acidic it is, right? Typically the titrant, the known solution, is added from a burette, this thing, to a known quantity of the analyte, the unknown solution. So you know the quantity of the analyte, which is at the bottom here, to the solution until the reaction is complete. So right when you have a different color, for example, from the blue it turns into red, then you stop the titration. So no longer the titrate is pouring down into the what was it called? The known solution, the unknown solution here. You have the quantity. Was it analyte? Yes, perfect. I'm just trying to remember it, right? Memorize it. Cool. So you have known volume for both, but for the titrant, the concentration is known, but for the analyte, you don't know the concentration. So at midpoint, the pH equals pKa, and at the equivalence point, the pH equals Pi. So what does this mean? Let's look at do we have titration midpoint and versus equivalence point. There you go. From titration curve provided, identify the pH at the endpoint. So the pH at the endpoint is 8 here, as we can see, but let's look at this. Equivalence point is the point in the titration where neutralization occurs, the point where there are equal parts, and midpoint, the point in the titration where half the analyte has been neutralized. So in equivalence, it is completely neutralized, right? There are equal parts basically, right, of the titrant and the analyte, but in the midpoint is half of the analyte that has been neutralized. On the x-axis, this will be the halfway between the starting point and the curve and the equivalence point. Midpoint is also called the buffer region. What is the volume of NaOH added at approximate equivalence point? So from what was said, I'm assuming the equivalence point is here. Using definitions of equivalence point, find what is asked. At the equivalence point, pH should be neutral at neutral 7. So we want a 7 neutral point here. So we draw a line from 7 on the y-axis to the curve. And 30 is the answer, right? 29 milliliters, the volume at the equivalence point. Now analyze the following curve. What is the volume of HCl added at the approximate midpoint? Now we want the midpoint. So is it half of 7? We have to find equivalence point to find the midpoint. So we find the pH 7, it's 26. The equivalence point appears to be 26 milliliters HCl added. To find the midpoint, you just divide it by half, which makes it 13 milliliters. This is the buffer region or midpoint. So we can write here that the midpoint is also known as the buffer region. So at the midpoint, where it's half of the equivalence point, the pH equals pKa, right? Because pKa equals half of the when half of the solution is deprotonated. Now the equivalence point equals the Pi. So the solution is completely neutralized. It's completely in the Zweiterion form. Now let's continue. We have the peptide bond formation and hydrolysis. So the terminology is that dipeptides have 2 residues, tripeptides have 3 residues, oligopeptides have less than 20 residues, and more than 20 residues we have polypeptides. So we have to know that oligo is less than 20 and poly is over 20. Now the formation of the peptide bond, right? Forming a peptide bond is a dehydration reaction. This is important. It's also in blue, so we don't need to highlight it. And the nucleophilic amino group of one amino acid, so the nitrogen from that amino group, it is called nucleophilic. So it likes to attack a nucleus, right? A proton. So attacks the electrophilic carbonyl group of another amino acid. So the carbonyl on the carboxylic acid group is electrophilic. It likes electrons, and those electrons are coming from the nitrogen that is nucleophilic, and it likes the nucleus, the hydrogen on the carbonyl group, right? That carbonyl group is electrophilic, so it likes electrons. We can ask, like, why does the carbonyl group, Y is the carbonyl group, is it its electrophile. Carbons have a resonance contribution that places a positive charge on carbon and a negative charge on oxygen, making the carbon essentially permanently electrophilic. Because it's positive, that carbon, it is completely electrophilic. It likes electrons. The opposites attract, right? So nucleophilic, amino, electrophilic, carbonyl. Amide bonds, it's this carbon-nitrogen bond of a peptide bond. So the peptide bond that forms this interesting. So it's also rigid due to resonance. So this carbon-nitrogen bond is called an amide bond or a peptide bond, right? So the peptide bond is basically an amide bond, amide, peptide. So we can call amides in order to understand peptides better, right? The pep comes from the protein part, probably. So the carbon and nitrogen, when this forms, when the nucleophilic amino group attacks the electrophilic carbonyl, the carbon, so that formation of the bond is called the peptide bond, or it's an amide bond, and it's rigid. It's strong due to the resonance. Breaking a peptide bond is a hydrolysis reaction. So when you break the peptide bond, you need water. So you need the oxygen probably to attack the carbonyl, that carbon, and replace the nitrogen. So you need to break your proteins with water. That's why we actually probably shake our protein shakes, right? In order to dissolve it in the water or the milk or the liquid, in order to be able to ingest it, make it liquidy, not rigid, which is caused by the resonance in that structure of the amide bond. Nice. Okay, I didn't mean to do this. Let me. Okay, I just want the hydrolysis to be highlighted. Perfect. And breaking. Now we have primary and secondary protein structures. So the primary structure is a linear sequence of amino acids in a peptide stabilized by peptide bonds. The amino acid sequence is written N terminus to C terminus, and we know that N terminus is positively charged due to NH3 plus. Yes. Now the secondary structure is the local structure of neighboring amino acids. It is stabilized by hydrogen bonding. So hydrogen bonding is a specific feature of secondary structures between amino groups and non-adjacent carboxyl groups. So it's not beside it, it's between the amino groups and between amino and non-adjacent carboxyl groups. Alpha helices, a common secondary structure. Clockwise coils around a central axis. So we have clockwise coils in alpha helices. I didn't know that it is clockwise. If they asked if it was anti-clockwise, is there another name for it? Clockwise and anti-clockwise? There was something else for anti-clockwise, right? I think I'm making up words. Yeah, correct me if I'm wrong, please. And we have beta pleated sheets. These are also common secondary structures. They are rippled strands that can be parallel or anti-parallel. So let me actually, the or is important. Proline can interrupt secondary structure because of its rigid cyclic structure. So proline is what interrupts secondary structures because of its rigid cyclic structure. Primary protein structure sequence of a chain of amino acids, again linear sequence, right, of amino acids in a peptide. So it's a peptide bond stabilized by peptide bonds. Yes, the amide bond between the carbon and the nitrogen. The amino acid sequence is written N terminus to C terminus. So we go, whenever you see an amino acid sequence, it's from N to carbon, it's from the amino group to the carboxylic group, and N terminus is positively charged due to NH3 plus. And also the C terminus is negatively charged due to the CO minus. Then for secondary protein structure, local folding of the polypeptide chain to helices or sheets. And then we have the tertiary protein, where three-dimensional folding pattern of a protein due to side chain interactions. So now we come with the side chain interactions. And quaternary structure is protein consisting of more than one amino acid chain. Note, denaturing is when a protein or nucleic acid loses its quaternary, tertiary, or secondary structures due to breaking non-covalent interactions such as hydrogen bonds, hydrophobic interactions, and dipole-dipole interactions. So when you break non-covalent interactions such as hydrogen bonds, so hydrophobic interactions, hydrogen bonds, and dipole-dipole interactions are a subtype of non-covalent interactions. What are covalent interactions? I think it's ions, right? So any covalent interaction is not accepted, and it's not part of denaturing of the quaternary, tertiary, or secondary. So yeah, and for the secondary, we need to know that hydrogen bond is primarily dominant in those secondary structures. Now the tertiary structure, it's a 3D shape of a single polypeptide chain, and it is stabilized by hydrophobic interactions, specifically acid-base interactions, specifically H bond, hydrogen bond still, and disulfide bonds. This is when we get the cysteines, right? They have sulfur. Where is the acid-base interactions from? We have 3 acids and 2 bases. Or it's the opposite, sorry. We have 2 acids, which are aspartate and glutamate, and 3 bases that are lysine, arginine, and histidine. Then we have hydrophobic interactions. So you push hydrophobic R groups to the interior or a protein, which increases entropy of the surrounding water molecules and creates a negative Gibbs free energy. So hydrophobic interactions, what are they? Okay, let's not do that. Hydrophobic, push hydrophobic R groups. So we have like 6 or 7 of them. I have to redo my amino acid revision, but they push hydrophobic R groups into the interior of the protein of the tertiary structure, or a protein which increases entropy of the surrounding water molecules. So what does this mean? The protein increases entropy to the interior, or a protein. Okay. This hydrophobic interaction increases entropy of the surrounding water molecules and creates a negative Gibbs free energy. So increases entropy of surrounding water molecules and creates negative Gibbs free energy. So when we have negative Gibbs free energy, it's stabilized. An increase of entropy also causes stabilization, right? And this is why the 3D shape is stabilized by hydrophobic interactions. We also have disulfide bonds occurs between the two cysteine molecules that are oxidized and create a covalent bond between their thiol groups. Covalent bond. This is ionic bond, right? So disulfide bonds are ionic, covalent actually. This forms cysteine, like, so these thiol groups form covalent bond, and the cysteine molecules are oxidized. When you're oxidized, you're losing electrons. So the sulfur is losing electrons. Those electrons are being attached to oxygens. So then you get the thiol groups, which make the covalent bond, and this forms cysteine, which is an amine, not an ene. There should be some differences here, right? We can see that probably in our flashcards. Then we have quaternary structures, which are, but still we didn't talk about the acid-base interactions and hydrogen bonds in 3D shape proteins, which we can come to that later. And then we have quaternary structures, which is the interaction between peptides in proteins that contain multiple subunits. So these quaternary structures are formed by the interaction between the peptides in proteins that contain multiple subunits. So now we have multiple subunits of these 3D shapes. And we have conjugated proteins, which are proteins with covalently attached molecules. So again, we are facing here with a covalent bond that makes conjugated proteins. We'll come to that in the flashcards, hopefully. If not, we can just note it down. So I'm writing down cysteine, and conjugated proteins. We also have prosthetic groups. So for example, just back at conjugated proteins, the disulfide bond, which is a covalent bond, should be considered a conjugated, like in the conjugated proteins, right? You have prosthetic group, which is the attached molecule in a conjugated protein. So that attached molecule is the conjugated protein. Can be a metal ion, vitamin, lipid, carbohydrate, or nucleic acid, right? So cool. So we have metal ion. Should I color them, color these examples with a different color? Metal ion, vitamin, lipid, carbohydrate, or nucleic acid. No. Let's not do this. Oops. Denaturation is the loss of the 3D structure, but still denaturing, we saw that it happens for the secondary structures too, with the hydrogen bonds that can be broken, and it is caused by heat. Let me actually go with yellow again. heat or solute concentration. So like acid base probably. And this is the polypeptide structure. This is the peptide bonds here between the nitrogen and the carbon, or also these are amide bonds. And in order to break them, you need hydrolysis, and vice versa. When they form up, you have dehydration. Perfect. I'm just gonna take a break, and I'll see you tonight. Probably I'll go do the flashcards tonight because I'm going out right now. Peace.